In today's video, we will be talking about two important topics in food science and technology, which is quality analysis of adulterants and isolation of food spoilage microorganisms. So we will begin with isolation of food spoilage microorganisms. Now, first, we'll begin with what is food spoilage. Food spoilage is defined as a deterioration of food quality. It is caused by It is caused by the growth of bacteria, yeast, as well as molds. What happens to the food when it is getting spoiled? It changes in texture, taste, odor, appearance. Now, when we isolate the food spoilage microorganisms from the food samples, we can identify what are these microorganisms that are causing this spoilage. We can understand what are their growth conditions, and all this will help us to prevent and preserve the food from getting spoiled. Now, when we talk about the definition of food spoilage, the definition of food spoilage is defined as a metabolic process that causes foods to be undesirable or unacceptable for human consumption. Now, food spoilage can be caused from in the food items by two different types of factors. The intrinsic factors include the changes in pH, water activity, nutrient content, oxidation reduction potential, and antimicrobial property. All these factors are going to increase the growth of these microorganisms, thus leading to spoilage of food. Whereas extrinsic factors are the factors coming from outside the environment or in the surrounding of the food, where we talk about temperature, humidity, and pressure. Apart from causing these spoilage in food, these microorganisms that are present in food can also cause foodborne diseases. These diseases are the type of diseases that can be transmitted when consuming the contaminated food items. We will now talk about the different type of microorganisms present in food and some common examples and how they affect the food items. Now, starting with bacteria, bacteria grow in neutral pH. Now, we can also have some beneficial bacteria. These beneficial bacteria are going to help in the process of fermentation. Examples, we have gram-positive examples. We have Bacillus, Staphylococcus, and Clostridium. Lactic acid bacteria is one common bacteria that's present in our milk products. Next, we have gram-negative bacteria. Examples are Salmonella, Shigella, E. coli. Now, what do these microorganisms do to the food? They cause off odors, they cause gas production, they cause discoloration, as well as changes in pH. And consumption of the food items that contain these pathogens will lead to foodborne illnesses. Moving to the next type of microorganism, we have molds, which is a type of fungi. They grow aerobically under low water activity and they can also grow in acidic pH. The type of microorganism here, we have Aspergillus, Penicillium, and Rhizopus as examples. They cause discoloration, they cause rotting, and they cause mycotoxin contamination. Mycotoxins are the toxins produced by the mycoclass, which is the fungus. Coming to yeast, yeast are unicellular fungal organisms. They prefer acidic pH. They grow in the food samples that contain high salt and sugar. Examples, we have Candida as well as Saccharomyces. They are most importantly used in fermentation. Therefore, we can find them in pickles, jams, sauces, and syrups. Moving forward, the protozoans, the next type, they are the single cell eukaryotes. They are mostly examples. We have Entamoeba histolytica and Toxoplasma gondii. These cause foodborne illnesses such as amoebiasis and toxoplasmosis. Next type of microorganism, we have algae. Algae are aquatic photosynthetic organisms. Examples, we have Microcystis aeruginosa and blue-green algae. They cause algal poisoning. Viruses, these are obligate intracellular parasites. They act as carriers and they are acid resistant. Examples, we have hepatitis and rotavirus. These cause foodborne viral infections like hepatitis and gastroenteritis. Moving to the next one, we have prions. Prions are misfolded proteins. They're mostly presented in meat. They're mostly present in meat. So when we don't cook meat properly, they have the probability of containing these misfolded proteins that can cause infections. We have bovine spongiform encephalopathy as well as the scrapie as examples. They cause neurodegenerative disorders and they're mostly present in contaminated meat. So this is a small overview of what are the types of microorganisms that can be present in the food samples that can cause spoilage. Now moving forward to the steps of the methods we can use for isolating food spoilage microorganisms. The first step is sample collection and preparation. We're going to collect the samples. If our food sample is a solid substance, we're first going to convert it into a slurry or a liquid substance by using a diluent. The diluent could be peptone water or buffered saline, where we're going to homogenize the sample and then we're going to use them for our isolation. Next is, if our sample is a liquid, we will be directly using them for the isolation. Step 2 involves serial dilution, where we're going to take our samples and we're going to dilute them in tenfold using our diluents. So whatever diluent we are using in the previous step will be used here itself. Now, why are we doing this process? We're doing this process in order to reduce the microbial load that is present in the food sample. Now, when we isolate these microorganisms on the nutrient plates, we don't want to see a lawn of bacteria. We want to see individual colonies, and that will happen when we do serial dilutions. So in serial dilution, the stock solution in our case, which is a food sample, is mixed with 900 microliters of the diluent. We will thoroughly mix this first sample, which has a dilution of 1 is to 10, and we will take 100 microliters from here and add it to the next 900 microliter of diluent. So what happens here, we are diluting the microbial load tenfold. So this will be 1 is to 10 power 2, 1 is to 10 power 3, so on and so forth. When we plate the dilutions in different agar plates, we will be able to see that the first diluent will have a lot of colonies, will not be able to differentiate anything, whereas the last diluent will show individual colonies which we can easily pick and continue our process of identification. Now, how do we plate these? Now, after we have serially diluted the samples, we have to plate these microorganisms onto agars. These agar are nothing but a nutrient media which are going to enable the growth of these microorganisms in vitro conditions, which is nothing but our lab. We could do pour plate method, spread plate method, and streak plate method. So in pour plate method, we are going to take an empty plate, we're going to add our agar, and then we're going to swirl to mix it. Whatever sample we have, which is our inoculum, is going to be present in the plate. Whereas in spread plate method, we'll have a drop of our sample onto pre-solidified agar and spread it to observe the growth of the microorganisms. In streak plate method, we are going to be using an inoculation loop. We are going to be picking up the sample and then streaking onto the plate in the direction that is shown in the picture. The use of this is to observe the discrete colonies. We can see individual colonies in the fourth quadrant. Now, when we want to grow specifically the spoilage microorganisms, we can use selective or differential media, which means we will use specific media for the growth of specific type of microorganisms. We can use McConkey agar for the growth of coliform bacteria, eosin methylene blue for the growth of E. coli, potato dextrose agar for the growth of molds and yeast, MRS agar for the growth of lactobacillus, and thioglycolate medium for Clostridium. So you can learn a few examples of the different type of selective media that we can use for the growth of specific microorganisms. So what does the selective media do? They give the nutrients that can specifically enable the growth of these particular microorganisms from the large variety of microorganisms present in the food sample. So from my food sample, when I plate my entire sample onto McConkey agar, only the coliform bacteria will grow in that agar because it is favoring the growth of these specific type of bacteria. Once we have grown these bacteria, we can also do enrichment. That is, before we add our samples to the plates or after we have isolated pure cultures, we can enrich the cultures. We can cause more amount of the culture to be present by using enrichment techniques, where we're going to inoculate our food samples in cooked meat broth or selenite F broth or tetrathionate broth for different types of microorganisms. What happens in enrichment is the number of microbes becomes more. So when more number of microbes are present, I can easily isolate them and identify them. For fungi and yeast, as we have already mentioned, we can use potato dextrose agar. We can also use the STA agar. For fungi as well as yeast, the incubation condition will be 25 to 28 degrees Celsius for 3 to 5 days. For anaerobic isolation, we use specialized equipment such as the anaerobic jars. We can also use a different kind of media such as anaerobic blood agar or thioglycolate broth. Now, after we have identified the cultures, now how do we purify them and how do we, after we've isolated the cultures, how do we purify them and how do we isolate them? For purification of cultures, we're going to pick single colonies and then try to grow them again using quadrant streak method. What happens here? We have individual colonies being growing in multiple plates. These are known as pure cultures. And how do we identify what type of microorganism this is? We can use three different types of methods. In microscopy, we can use gram staining, spore staining, and motility. Biochemical test will measure the type of enzymes released by the living microorganism, which will help us classify this particular organism present on the plate. Example, catalase, oxidase test, fermentation test, enzyme activity test can be used to identify what type of microorganism is present in the plate. Next, we can also use molecular methods where we're going to see the DNA of the microorganism using sequencing methods such as PCR for their identification. Now, why do we do identification of food spoilage microorganisms? When we identify this, we can identify the source of these contaminations. We can also enable better quality assurance. We can improve the shelf life of the food. When we try to identify what microorganisms are present, we can try to prevent it. Now, we are also going to be using these microorganisms to produce certain important enzymes and also for the biofilm formation in the equipment. So all this can be studied when we isolate these microorganisms and we explore how can it be prevented. Now, moving to the next topic, which is quality analysis of adulterants. We will first start by talking about what is adulteration. So, adulteration is addition of a certain substance which is going to be causing the source of the food to become less important. It's going to reduce the vital importance of food. So, let's say we are talking about tea. We have lots of different varieties of tea. Some herbal teas are said to improve our health. They can regulate our mood. Some herbal teas nowadays they come up with the idea of reducing headaches and releasing overall improving the mental perspective of a person when you drink such hot beverages. But now we know that the companies that sell these tea powders or these tea leaves are not doing justice. They can add some components into the tea packets which are not tea leaves. So such type of additions will reduce the vital importance of the food. And this substance that we are going to be adding, we call it as adulterant, and the process is called as adulteration. Here we have some examples and the types of diseases that these. So the most important is anemia, abortion, paralysis, brain damage, and cancer. Now, we are going to be talking about what is food adulteration. So, food adulteration is the intentional or accidental addition of inferior, harmful, and non permitted substances into food for the purpose of shelf life or profit. Now, adulteration will affect the nutritional value, it will affect the safety, it will affect the quality of the food, and also cause economic fraud and public health hazards. So, now how do we identify these adulterants from the normal food items? These adulterants will have a change in their chemical, physical, or their microscopic analysis can be done in order to differentiate between what is an adulterant and what is a food substance. So this is all done in food testing laboratories that are approved by FSSAI in accordance with the Codex regulations. Now, first we are going to see what are the adulterants in milk. We have starch, urea, detergents, formalin, as well as water as an adulterant in milk. So, starch can be identified by adding iodine to it. When we add iodine into the milk, if it contains starch, it will turn blue. Urea can be identified by adding bromothymol blue. The development of bluish green color indicates the presence of urea. Detergents can be identified by shaking the milk sample. If they have a soapy lather, they're going to indicate the presence of detergents. The next adulterant is formalin, which is added as a preservative. The addition of H2SO4 along the sides of the test tube will lead to the formation of violet or blue ring if there is a presence of formalin. When more amount of water is added, we can identify it using a lactometer. The next is the adulterant in oils and fats. Mineral oil can be identified by shaking it with concentrated sulfuric acid. If they turn red or brown, it indicates that mineral oil is present in a sample. Argemone oil can be identified by using nitric acid, and castor oil can be identified by again adding H2SO4. This reddish brown color formation will indicate that our oil has been adulterated. And vanaspati is a major adulterant that is added to butter and ghee. It can be identified by treating it with hydrochloric acid. Pink or red color formation leads to the identification of vanaspati in butter and ghee. Now, moving to the adulterants and spices. The turmeric powder is the first spice. The adulterant is lead chromate. We're going to add HCl. The effervescence or the red color will indicate the presence of lead chromate. Moving to the next one, we have black pepper. In papaya seeds act as a adulterant in black pepper because of their similarity. When we add them to water, the seeds will float, whereas our black pepper will be settled. So then we can identify that a pepper is being adulterated with papaya seeds. Moving to chili powder, nowadays companies are mixing brick powder or artificial colored powder in order to resemble chili powder. We can identify them by sprinkling it on water. The brick powder will settle down, the artificial color will be dissolved. Cumin seeds, the adulterant we're talking about is grass seeds. They can be differentiated by sprinkling on alcohol. The grass seeds will lose their color. Moving to the adulterants in sugar and jaggery, we have chalk powder, methanol yellow, sodium bicarbonate, and sand. Chalk powder can always be identified when we add acids. Chalk powder is a calcium compound. So when we add an acid, it will release carbon dioxide, which confirms that the effervescence will confirm that there is chalk powder present in my sugar and jaggery. Methanol yellow, which is a non-permitted food coloring substance, if it is present in a sample, we can identify it by using dilute HCl. The pink color formation indicates that methanol yellow is present in the sample. Sodium bicarbonate, which is washing soda, can also be added as an adulterant. I can identify it by using an acid such as HCl. The effervescence that arises when we add acids indicates the presence of washing soda. Moving to sand, when we dissolve it in water, the sand will settle down in the bottom, whereas our sugar and jaggery will be dissolved. Moving to the next adulterant, we have pulses in cereals. Kesari dal can be identified by visual examination. Artificially colored pulses, we can wash them with water. Chalk, again, we can use acid, and effervescence will indicate that there's presence of chalk. Iron filings can be identified by passing magnets over it. The iron particles will stick to the magnet. Next, we have the adulterants in tea and coffee. Iron filings can be identified by using a magnet. The exhausted tea leaves can be repacked again and sent as new fresh tea leaves. We can identify it when we sprinkle water on filter paper. The colored spots will indicate that there is artificial coloring. The talc or soap stone that can be added in coffee, we can dissolve coffee in water. The soap stone will settle. Chicory is a adulterant in coffee, although some amounts of chicory is now used in order to give the taste or the particular aroma for the coffee. So chicory in coffee can be sprinkled on water. Chicory will settle off faster than coffee. Now the adulterants in salt, we have chalk powder and powdered glass. So powdered glass is a very dangerous adulterant present in salt. It can be identified by the microscopic examination. So these powdered glass items will resemble the crystals of salt, but they will not, our salt molecules will not be able to reflect light, whereas glass molecules will be able to reflect light. Chalk powder, again, we can add HCl or any type of acid, and effervescence will indicate chalk. Now, why do we do qualitative analysis for food adulterants? We do this process in order to make sure that safe food is reaching the public and therefore public health can be maintained. We can prevent economic fraud. So all these companies are adding these adulterants in order to sell less amount for more money. They say the quality has been improved, but what they're adding is the adulterant. So economic fraud can be prevented. It will help us find out if the industries are meeting the standards of FSSAI, which is a major institute that has laid the rules. It helps in increasing consumer awareness. It helps in quality assurance in the food industry, and it will also help us to work on our research, which helps us on developing better rules and better methods to identify these adulterants. So, with this, we come to the end of the topic. Thank you so much for listening.